Hello guys, welcome to my channel. So as you know, if you saw the previous videos that heparin sulfate, right? We are studying over the polysaccharide and specifically the heparin sulfate. If you remember, like we studied about the NS like this and this and this, for example, and the dark one is the NS and this is the N-domain of the heparin sulfate molecule, right? So the protease thrombin essential to the blood coagulation is inhibited by another blood protein antithrombin which prevents premature blood clotting, correct? Alright, so the antithrombin does not bind to or inhibit thrombin in the absence of heparin sulfate. So antithrombin will not bind the thrombin, right? So antithrombin and this is thrombin. Right, so in the absence of heparin sulfate, so antithrombin does not bind to the thrombin, right? So in the presence of heparin sulfate or heparin, the binding affinity of this antithrombin increases 2000 fold. So in the presence of heparin sulfate, or you can call heparin, right? So the antithrombin binding affinity, so I'll just write 80. Antithrombin binding affinity increases 2000 fold in the presence of heparin sulfate, right? And the thrombin is strongly inhibited, right? So when thrombin and antithrombin are crystallized in the presence of a short segment of heparin sulfate, the negatively charged heparin sulfate mimic is seen to bridge positively charged region of two proteins, causing an allosteric change that inhibits thrombin's protease activity. And so what happens? So when the thrombin and antithrombin are crystallized in the presence of a short segment of heparin sulfate. So for example, this is like a short segment of heparin sulfate, right? Na and NS and right, NSNA. Right, this is a short segment of heparin sulfate, for example. That is a short 16 residue segment. So the negatively charged heparin sulfate. So this is a negatively charged, right? So as it has carboxylic group and other groups that is strongly negative. So it will mimic is seen to bridge positively charged. So the negatively charged, this will attract the AT, and for example this attract the T, that is thrombin TH, right? It will both attract the protein, right? And then it will try to connect each other. It will act as a bridge, right? That will cause an allosteric change and that will inhibit thrombin's protease activity, right? When the antithrombin is acting on thrombin. So the binding site of heparin sulfate and heparin in both proteins are rich in, so heparin sulfate or heparin, so both are rich in ARG and LYS residues, lysine and arginine. These are the amino acid residues. So the amino acid positive charge interact electrostatically with the sulfates, right? This and interact with the negatively charged with the sulfates of the glycosaminoglycans, and the antithrombin, right, inhibits thrombin protease activity, and antithrombin also inhibits two other blood coagulation proteins. So antithrombin inhibits two other blood coagulation factors. So which blood coagulation proteins are proteins? That is factor 10A and factor 9A, right? 9 and 10 right? So antithrombin also inhibits these two other blood coagulation factors in an heparin sulfate dependent process. So this process of inhibition is also dependent on the heparin sulfate, right? So this means that the heparin sulfate is very important, and the synthesizing of the sulfate domains are also really important in heparin sulfate. So how it is demonstrated, the importance of the correctly synthesizing sulfate domains in heparin sulfate is demonstrated in the mutant or knockout mice lacking the enzyme that sulfates the C2 hydroxyl of iodinoate, right? It does not have that enzyme. So these animals that does not have C2 hydroxyl of iodinoate, it they are born without kidneys and with severe developmental abnormalities of the skeleton and eyes. So and other studies demonstrate that the membrane proteoglycans are important in the liver for clearing lipoproteins from the blood, right? So the proteoglycans are also really important, right? So the study shows that it will help liver for clearing the lipoproteins from the blood. And finally, there is growing evidence that proteoglycans containing heparin sulfate and chondroitin sulfate provide directional cues for axon outgrowth, influencing the path taken by developing axons in the nervous system. So this is why the proteoglycans is important. Right, so there is one more kind of proteoglycan. So some proteoglycans can form a proteoglycan aggregates, enormous supramolecular assemblies of many core protein all bound to a single molecule of high aileron. So for example, aggregan, aggregan. So this is aggregan core protein. So this is a core protein, right? Aggregan. I'll write it down. Core protein, right? Aggregan core protein. This has a multiple chains of chondroitin sulfate and keratin sulfate that is joined to serine residues in the core protein right through the trisaccharide linkers to give an aggregan. The monomer of that is the monomer is M and 250,000, right? So that's the molecular weight of the aggregan core protein. So how this proteoglycan aggregates is actually occurring. So as as I mentioned, so the proteoglycan aggregates is enormous supramolecular assemblies of many core proteins. So this is a one kind of core protein that is all bound to a single molecule of high allogenome. So for example, this is the molecule of high allogenome. So it is bound to the core protein, and the core protein is bound to the hyaluronate, right? And it has, so this aggregate core protein has a multiple chains of chondroitin sulfate and keratin sulfate. So all right, let me take the brown one. So for example, this is chondroitin sulfate and the brown ones are keratin sulfate, right? Right, so this is something like this. So this is how it is, the aggregate core protein, right? All right, so we'll just tag this. Hello. And this is core protein. That is this aggregan, and the brown, for example, the black one is chondroitin sulfate, and this one is keratin sulfate, right? Sorry. So something like this. So when 100 or more of this decorated core protein bind a single high alluronate, extended molecule of this high alluronate, the resulting proteoglycan aggregate, so this is a proteoglycan aggregate, and its associated water of hydration occupy a volume about equal to that of a bacterial cell, right? So aggregan aggrecan interacts strongly with collagen in the extracellular matrix of cartilage, right? So this aggrecan can interact strongly with the collagen in the extracellular matrix of cartilages, contributing to the development, tensile strength, and resilience of this connective tissue. So this is how it gives the tensile strength, resilience, and development to the connective tissue. Right, you can see a picture on your screen, so I will explain to you. So the interwoven with this enormous extracellular proteoglycans are fibrous matrix proteins, such as, so like with this, the one that you are seeing in the picture, so like there is an interwoven fibrous matrix proteins as well, that is collagen, elastin, and fibronectin, forming a cross-linked meshwork that gives the whole extracellular matrix strength and resilience. So some of these proteins are multi-adhesive, right? A single protein having binding site for several different matrix molecules. Fibronectin, for example, has separate domains that binds fibrin, heparin sulfate, collagen, and a family of plasma membrane proteins called integrins that mediate signaling between cell interior and the extracellular matrix. So this is how just one fibronectin can work. Just one single protein having several different matrix molecules or different binding sites, right? So the overall picture of cell matrix interaction that emerges shows an arrays of interaction between the cellular and the extracellular molecules. So this is how it is connected. This interaction serves not merely to anchor cells to the extracellular matrix, but providing the strength and elasticity of skin and joints. Right? So this helps in the strength and elasticity of skin and joints, and they also provide the path that direct migration of cells in developing tissues and serve to convey information in both directions across the plasma membrane. Right, so that was proteoglycans, and this is how it helps, right, in our body. All right, guys, thank you guys for watching, and we'll see you in the next video with the next topic.